It's my pleasure and indeed an honor to introduce our first speaker, keynote speaker, Professor Cynthia Wolberger, who is at the Johns Hopkins University School of Medicine, where she's recently become a department chair. She's a leader in the field of DNA packaging and transcription regulation. And the title of her keynote presentation today is Histone H4 in transcription regulation. Thank you very much, Cynthia. Welcome, and we thank you for your longstanding service as an advisor and chair of the advisory committee to RCSB Protein Data Bank.
Thank you very much for the invitation. And really, before I begin my talk, I just want to comment on this incredible milestone, the 50th anniversary of the PDB. As someone who began their training 40 years ago, when the PDB still had a small number of structures, and equally as importantly, there was no mandate to deposit structures, it's really hard to describe to the younger scientists today just how transformative it has been and how much it has really fueled the growth of structural biology and the utility of structural biology to have so much structural information available as a PDB around the world. And not only that, but to have it accompanied by all of these tools for analyzing structure and correlating it with all sorts of other data that we have from protein sequences, DNA, and biology. And that I want to particularly call out Helen Berman, who had the vision to transform a PDB from a simple depository in which one deposited and then withdrew structures to one where really we could analyze and sort of take it to the next level. And as Stephen said, it's really been a guiding light and a model for all other databases in biology. Okay, so with that, I'd like to start and talk about some recent work that we've been doing in the lab. So someone confirm that this is showing up okay? Yes? Okay, I don't hear anyone. Please shout if something's wrong with that.
No, we don't see it.
You don't see it. Aha,
No,
thank you.
Yet.
Because I didn't click that critical share screen. Thank you. Okay. All right. Now we're good. All right, great. And I've got to shrink the little, okay. All right, so as Stephen said, I'm going to be talking with work we've done in the lab on histone H2B-ubiquitination in transcription regulation. And so in each of our cells, there's about 2 meters of DNA, and that is organized and tightly packed into the nucleus by the histone proteins into this nucleoprotein complex called chromatin. And the fundamental organizational unit of our chromatin is the nucleosome. And the nucleosome consists of a protein core with 2 copies each of histones H2A, H2B, H3, and H4 of about 146 base pairs of DNA wrapped twice around that core. So pretty much every process that involves the cell machinery gaining access to that DNA is regulated by post-translational modifications that are deposited on specific residues in each of the 4 core histone proteins. And there's an enormous chemical variety of these modifications. And it's the particular type of modification, as well as the particular residue in the particular histone that is modified, that is important for regulation. And what I'll be focusing on in my talk are the histone modifications that are involved in regulating transcription. So the modifications vary enormously, both in chemical nature and in size. So there are small chemical modifications such as acetylation, methylation, phosphorylation, and you see an example here of methylation of lysine. So here's an unmodified lysine side chain, and here's a lysine modified with a methyl group, all the way up to the attachment of entire proteins to a lysine side chain, in this case ubiquitin, which is a 76 amino acid protein that's covalently linked to lysine side chains via its C terminus, via an isopeptide linkage. Now, if you were to examine different regions of chromatin, and look at the post-translational modification, what one sees is that there's a very different pattern of post-translational modification in heterochromatic regions. So these are regions that are highly packaged and have little to no transcription, versus euchromatin. So this is a more open chromatin. So we call beads on a string. In an electron micrograph, you could see the individual nucleosomes, and these regions are actively transcribed. And there's a very different pattern of post-translational modifications that distinguishes them. So these different patterns of modifications associated with different chromatin states has been come to be called the histone code, which was a phrase coined by David Alice. And so what the histone code refers to is the particular array of modifications. And then these modifications are read by particular proteins. So they serve as landing pads for protein domains that recognize the specific modifications. And those domains are part of larger enzymes or enzyme complexes that are then recruited by those modifications. And then the modifications are regulated by the so-called writer proteins. So these are the enzymes that covalently attach the modifications, and then eraser proteins, so the proteins that remove the modifications. So the modification I'll be focusing on is ubiquitination of histone proteins and crosstalk between that ubiquitination and other modifications. So histone monoubiquitination plays a non-degradative role in gene regulation. You're all presumably familiar with the role of ubiquitination in targeting proteins to the proteasome for degradation, but that is not what I'm talking about. I'm referring to this as yet another regulatory modification. And histone ubiquitination does not have a particular outcome. It depends upon where the modification is. So modification of histone H2A at lysine 119 is a hallmark of heterochromatin, so silent regions of chromatin, whereas monoubiquitination of histone H2B is a hallmark of actively transcribed chromatin, and it actually also plays a role in DNA replication and DNA repair, as well as nucleosome dynamics. I'll focus on the transcription aspect of this. So if you look across an actively transcribed region, what you'll find is a gradient of histone H2B monoubiquitination. So peaking near the transcription start site, but extending over the entire transcribed region. Now, histone H2B monoubiquitination plays a number of different roles in transcription, but the important one that I'll be focusing on is the crosstalk between that modification and methylation. So monoubiquitination of histone H2B is required for deposition of two different methyl marks, which in turn are also important for activating transcription. One of these is methylation of histone H3 lysine 4, and actually up to 3 methyl groups can be attached, mono, di, and trimethyl. And I'm going to use this shorthand throughout my talk. So H3 refers to histone H3, K4 is lysine 4, and also I'll often gloss over exactly how many methyl groups. So there's histone H3 lysine 4, and that's particularly peaked near the transcription star site. And then histone H3 lysine 79 with di and trimethylation extends across the entire gene region. So the histone H2B monoubiquitination stimulates two very different histone methyltransferases to deposit the methyl groups. One of these is histone H3 lysine 79 methyltransferase, which is called Dot1. And it's called Dot1 in yeast, and then Dot1L or Dot1-like in humans. And then histone H3 lysine 4 methylation is mediated by compass. So this is a multi-protein complex in yeast, and the human homolog is called MLL1. These contain a catalytic domain that belongs to the SET1 family. Now, even though both of these complexes are stimulated by histone monoubiquitination, they are located in very different structural environments. So lysine 79 of histone H3 is located in the globular core of the histone octamer, and it's located relatively close to the H2B monoubiquitination site, whereas histone H3 lysine 4 is located in this flexible tail of histone H3 and is located rather far from the site. Could that microphone be muted? Thank you. So what I want to tell you about is work that we've done in the lab to understand the molecular basis of crosstalk between monoubiquitination of histone H2B and methylation of each of these two lysine residues. And I'm going to start by telling you about our work on stimulation of the histone H3 lysine 79 methyltransferase by human Dot1L. So if you look overall and group genes according to whether they are transcribed at a low, medium, or high level, what one sees is there's a strong correlation between relative levels of histone H3 lysine 79 methylation. So this is taken from this paper, shows this is low, medium, high. This is monomethylation, average across a transcribed region, a gene body, and then this shows di and trimethylation, which again is correlated with the relative levels of transcription. We still don't know exactly how this stimulates transcription, but it's very tightly correlated with the relative levels of transcription. So it was first shown in yeast and then subsequently in human cells that if you knock out the ability of the cell to monoubiquitinate histone H2B, so that could be either by deleting the enzyme that's involved, so RAD6 is an E2, or just mutating the residue that's monoubiquitinated from a lysine to one that cannot be ubiquitinated, you then knock out the detectable histone H3 lysine 79 methylation. So this is a Western blot, so no methylation if you knock out either of these. And this is the same experiment that is done, or comparable experiment done in human cells. So here in wild type cells are levels of monodi trimethylation. And now if you knock out the E3 ligase, you greatly decrease methylation of histone H3 lysine 79. So the Dot1L protein, which is the methyltransferase that depends on monoubiquitination of histone H2B, is about a 1,500 amino acid protein, but the business end is here at the amino terminus. So the catalytic domain is about 350 amino acids. Shown here is the structure of the human Dot1L protein. There are also structures of the yeast protein. They look virtually identical. And then there are other regions actually of unknown structure, DNA binding domain, and then an extensive region here that interacts with partner proteins that form higher order complexes with Dot1L. We know from biochemical studies from a number of labs that this piece alone can be stimulated by monoubiquitination of histone H2B. But one of the mysteries when we first started this project is it has no known ubiquitin binding motifs, and attempts to sort of introduce random mutations to try to knock out stimulation by ubiquitin were not successful. So one of the questions is, how does this stimulate it, but how does this even bind ubiquitin? Now, I couldn't resist showing that, as I think everyone knows, the AlphaFold 2 has burst on the scene, and the Google consortium has now created a depository. This is actually in collaboration with EBI in Europe, of the predicted structures of all these proteins. So we naturally couldn't resist looking at what they predicted for all the rest of Dot1L. And that's shown over here. So green is the catalytic domain over here, and we've got some interesting alpha helices up to here, and then the rest of it is mostly spaghetti. Anyway, I have nothing more to say about this except that this has been yet another exciting development in structural biology, and this is going to be a fantastic tool for us to build into cryo-EM maps and come up with new constructs to study in our studies of Dot1L and everything else we're studying in the lab. Okay, so in addition to being interesting just from a mechanistic point of view, Dot1L is actually a sought after drug target, because in MLL rearranged leukemia, in about 40% of cases, there are fusion proteins of the MLL complex, which is actually the H3K4 methyltransferase, and the catalytic domain gets replaced by a fusion partner of Dot1L, and then Dot1L is misregulated, recruited to genes where it hypermethylates histone H3K4, and then those genes are hyperactivated. So a side sort of interest in this is actually using our structural insights for drug discovery. Another question we had going into this was even how Dot1L got accessed its substrate. So if you look at the structure of the nucleosome, sorry, I didn't put I've been putting PDB codes and I neglected to put one AOI here, Luger Richmond structure. So if you look at the K79 side chain, it's actually not very accessible. It lies along the surface of the nucleosome, and that lysine side chain, if you take the structure of Dot1L and try to dock it into the active site coming near to the methyl donor, which is S-adenosylmethionine, you can't do that. So we decided to tackle how does Dot1L actually access its substrate, how does it bind ubiquitin, and could we understand something about why ubiquitin is stimulating it. And so to get at that problem, we determined structures of Dot1L bound to ubiquitinated nucleosomes. And this was work done by an incredibly talented postdoctoral fellow, Evan Warden, and he was helped with some of this by Nick Hoffman, another terrific postdoc, a former postdoc in the lab. And I'm not going to go into all the technical details there, and that this is published in our paper, but I'll say we first determined a structure of Dot1L bound to this nucleosome that turned out to be in what we call a poised state. And then using replacing H3K79 with norleucine, we were able to trap what is an active conformation of Dot1L bound to a nucleosome. And this was determined at a resolution of 3 angstroms. Now, this was the first cryo-EM structure in my lab. I am I guess at this point, old school X-ray crystallographer. We've done many X-ray crystal structures, but with the new capability to do cryo-EM, we ventured out and did this. And I have to say, at the time, I was a little bit of a skeptic about low resolution things until I examined the really stunning density maps that we got. So this is a 3 angstrom resolution. This is, and you could see the amazing electron density. So we could see S-adenosylmethionine, resolved DNA base pairs. We see side chains. So ever since then, like many crystallographers, we've become converts to cryo-EM. Okay, so what did we learn? So what I'm going to show you is that what we learned was that, well, we learned how Dot1L binds to ubiquitin. And we found that ubiquitin serves as sort of an anchor site, allowing Dot1L to pivot between these two states. And now I hope this movie will work in Zoom. Great, so we're going to dissolve away the active state map. And what I'm showing you here is a pivot between this poised state with the enzyme lifted up off the surface of the nucleosome and this active state over here. So here is this ubiquitin pivot. We don't see the connection because that density wasn't great, but it's holding on to ubiquitin and allowing it to pivot from a state up off the surface of the nucleosome to one that's down on the nucleosome where it can access its side chain. So here's, so Dot1L in green, ubiquitin is always in purple. So in this active state, we actually see there's a tail of one of the other histones, Histone H4, that's reaching up, and it looks as if it's sort of tethering the catalytic domain in the active state, helping to position it on the surface of the nucleosome. So this is not a tail that is modified, but a tail of the nucleosome that is disordered in the poised state, and in various crystal structures, it adopts either different conformations or you don't see it at all because it's variable. But here it's in a very defined position, holding on to the Dot1L enzyme. So I had said that Dot1L, we couldn't figure out how Dot1L even recognized ubiquitin, and it turns out that it just had a ubiquitin binding motif that wasn't like any other ubiquitin binding motif. So this helix over here, this actually is a rigid helix. It's not, if I filled in all the side chains, you would see. I think of it as sort of a hand at the end with a thumb sticking out, grasping onto ubiquitin. It doesn't change conformation, but has a hydrophobic cluster of amino acids that binds to this hydrophobic patch here in ubiquitin. And I'm not showing you any of the biochemistry in my talk, but all of this we've verified, of course, with mutations in Dot1L, and there are mutations in ubiquitin that support this model. Now, I showed you that there's this tail of histone H4 that helps to position Dot1L on the nucleosome over here. And then at this other end, we have this ubiquitin interaction and an interaction with the acidic patch of the nucleosome. So I already showed you that the enzyme is pivoting around ubiquitin, but a second part of the pivot point is an arginine side chain that is interacting. So the arginine is located back here and is interacting with this acidic patch in the surface of the nucleosome. So this is the electrostatic surface potential, and shown here in red is a conserved patch of acidic residues that one finds in all nucleosomes, and we have this arginine anchor that's anchoring it here. So with the electropositive anchored to this electronegative patch in the nucleosome, and that is the same contact in both the active and the poised state. So this is a second portion of what allows Dot1L to pivot between these two positions on the nucleosome. So one of the questions that I told you that we had was how Dot1L was even able to access its substrate lysine. So I told you that we weren't able to build any models that could take just the crystal structure and dock it on the nucleosome. And to be honest, we thought that there were going to be some significant conformational changes within Dot1L itself that would then allow that side chain to get up into the nucleosome. And so we were very surprised to find that it's actually the globular core of the nucleosome that changes conformation. So this is the conformation in the, oh no, don't tell me my movie's not working. Oh dear. Well, sorry, I had this very cool morphing movie, so you'll have to believe the morph. So that side chain is goes from the conformation lying down here on the surface of the nucleosome to a pretty significant conformational change that allows it, points it straight up and sticks it right in the active site. And now let's see if this movie works. Okay, that one worked. So what you could see from this movie here, and I'll play it again, is watch the domain here pivot down onto the surface of the nucleosome, but watch what happens over here with the side chain. It's a conformational change right in the middle of histone H3 that allows that side chain to swing up into the active site. So it swings up by about 90 degrees, and that the NH3 group moves by about 10 angstroms to go up into the active site. And now this is within striking distance of the methyl group where the chemistry can occur. So this was really incredibly exciting for us because the core of the histone octamer, and you take a tour through the PDB of the many, many nucleosome structures there now, is a rock. It doesn't change conformation. And so this was very exciting and surprising to see this conformational change in the globular core. And I'll show you a couple of other images, movies of that I hope will work. So what assists this conformational change? There are actually, we are breaking of hydrogen bonds in a helix, and it's a energetically costly conformational change to induce. And it's assisted by this tail of histone H4. So I told you a number of slides back that in this active state, this tail of histone H4 comes and binds to Dot1L, and it actually binds in this groove within Dot1L, but it doesn't only simply stabilize the position of that catalytic domain on the surface of the nucleosome, but it also assists in that conformational change. And now I really hope this movie works. So here is now lysine 79. There's the tip of the lysine side chain. And here is, so this is in the middle of the globular domain. We're looking at the end of the helix here of histone H3. And what I hope this animation will show is the pulling apart of that helix. So those hydrogen bonds are being broken as that side chain now swings up towards us. So that is energetically costly to induce that conformational change, to break those hydrogen bonds, but it's compensated in part by these interactions with the tail of histone H4. So this inserts an arginine residue in there, and these carbonyl oxygens, which are facing outward in this change conformation, are now interacting with the arginine side chain here, so helping to stabilize that as well as to promote that conformation. And of course, not surprisingly, a mutation of the side chain just pretty much abolishes the ability of Dot1L to methylate H3K79. And then here's just another view of this. So here's arginine 19. Here's that lysine 79 pointing up now. And then in addition to this interaction, there are a whole slew of other side chains, actually a lot of hydrophobic side chains that are helping to prop this up and stabilizing this altered conformation. Okay, so I'd like to move to talk about briefly at least, our work on looking at how histone H2B stimulates methylation of histone H2B monoubiquitination stimulates methylation of H3K4. So we know a little more about what methylation of histone H3 lysine 4 does, and that is there are a number of complexes that are involved in activating transcription that contain within them domains that bind preferentially to H3K4 when it is methylated, and that interaction helps to recruit these complexes. One of them is the SAGA complex, which is a giant coactivator complex, and methylation of this residue actually stimulates yet another modification, the histone acetylation by the GCN5 acetyltransferase, and that acetylation in turn helps to open chromatin and stimulate activated transcription. The methylation also helps to recruit CHG1, so chromatin remodeler that helps to move around nucleosomes, as well as a component of the transcription pre-initiation complex. So the enzyme complex that we did the structure of, and that I'll talk about, that methylates this and is stimulated by histone H2B monoubiquitination is called COMPASS in yeast, and the human homolog is MLL1, and there's an MLL2 complex too. So at the core is a catalytic domain, SET1, and there's a SET1-like domain here too, associated with quite a number of other subunits. And it had been shown by a number of labs, particularly Ali Shalati, that a subcomplex of this is the minimal complex required for full activity, as well as the ability to be stimulated by ubiquitination. And there's a structure of the Cerevisiae complex that was done by cryo-EM by Yorgos Skiniotis in collaboration with Ali Shalati. And then there's another, the K-lactis structure was done by Ning Zheng's lab. So we using the approach we use for Dot1L, took this minimal ubiquitin sensing complex and created complexes with monoubiquitinated histone H2B and determined the structure by cryo-EM. And this was done at overall resolution of just about 3.4 angstroms, and this shows the complex colored according to the local resolution of the complex. And this is again work by Evan Warden, the postdoc in the lab who worked on Dot1L as well. So this is quite different from Dot1L. So now this is really a pretty big beast. So a large complex that's spanning the surface of the nucleosome. Here's ubiquitin over here. In green is SET1, the catalytic domain. And there are a series of other domains that are mediating contacts with the DNA, as well as with other histone proteins. Now, unlike Dot1L, there's a significant conformational change that occurs in the APO complex that was determined. So this is from the cryo-EM structure from Yorgos Skiniotis' lab. And the complex, this is the Cerevisiae complex bound to the ubiquitinated nucleosome. And what I'm going to show you is a morph between the isolated COMPASS complex and the one we see bound to the nucleosome.
You have 5 minutes, Cynthia.
Okay, I'll move right along.
Thank
Okay,
you.
so this is, okay, good. So this is the morph of the complex. So you could see that in binding to ubiquitin, it folds around the nucleosome. And importantly, there's formation of this helix over here that we do not see at all in the apo complex. So this had been identified in Bob Rader's lab as a so-called RXR helix that was essential for activity on nucleosomes and the ability of the complex to be activated by H2B monoubiquitination. So I'll show you the conformational change here, which is pretty dramatic. So here's the apo complex, and we're looking at a helix here. This is not the set1 helix, sorry, the RXR helix. And what we're going to see is this extends into a beta strand now and nucleates formation of this alpha helix here, this RXR helix. And this, what's critically important is part of this is being contacted by ubiquitin. And so there's a hydrophobic patch here forming interactions with the ubiquitin. And the other portion of the helix that's nucleated is again forming these electrostatic contacts with this negatively charged patch on the nucleosome here. In addition to that, there's another conformational change in this other subunit here, SWD1, that also forms a hydrophobic patch. And that in turn binds to a hydrophobic patch on the surface of ubiquitin. So so in brief, so what does ubiquitin do, or how is it recognized? So in contrast with Dot1L, with this rigid recognition domain, there's a whole series of conformational changes that are involved in recognizing ubiquitin. There's this helix that forms of the RXR helix here, bridging contacts between ubiquitin and the surface of the nucleosome. And then this domain here, SWD1, this subunit, has a region that undergoes a conformational change that binds to another hydrophobic patch on the nucleosome. So what are the themes in comparing these two very different enzymes and how they're stimulated by ubiquitin and to methylate residues? And they are really surprisingly different. So in Dot1L, what it looks like is that ubiquitin restricts a conformational search of the enzyme, changing it, basically anchoring it at one end and greatly reducing its conformational freedom, so increasing the rate of methylation of H3K79. Whereas in the case of Compass, the ubiquitin is really helping to nucleate formation of that helix that in turn plays a critical role with recognizing the surface of the nucleosome. So with just a couple of minutes left, I wanted to just touch very briefly on some of our efforts to take our structural insights and use them in drug discovery. And what I'll actually show you is yet another complex, and that is one that's involved in regulating levels of H2B ubiquitination. So during transcription, the H2B, there's one set of enzymes that ubiquitinate H2B, and there actually are two different deubiquitinating enzymes in yeast that remove the H2B, and a larger number of H2B specific DUBs or deubiquitinating enzymes in humans. And one we've been focused on is the subcomplex in the SAGA complex, which in addition to acetylating histones during transcription, also removes monoubiquitinated H2B. What I'm showing you here is the structure of the yeast deubiquitinating complex. The green is the catalytic domain, and it has three other proteins that it associates with. UBP8 is the yeast protein, USP22 is the human. And this is an important drug target because it's part of a death by cancer signature. It is hyperactivated in a variety of cancers that are refractory to pretty much any treatment. And based on a number of studies we had done in solution and structural studies, we had a hypothesis that a peptide-like molecule could be an effective inhibitor of the DUB module. That hypothesis turned out to be wrong, so I won't even take you through all the structures and the reasoning, but we were still able to find a peptide-like inhibitor. And we did that by collaborating with Hiro Suga at the University of Tokyo, who has this amazing platform of 10 to the 12th combinatorial cyclic peptides that are each tagged with the sequence, with the DNA and RNA sequence that encodes the peptide. And he has a selection method where you have the pool and you anchor your substrate to magnetic beads, and you go through rounds of selection looking for tight binders. And then Mike Morgan in the lab took over from there and tested those tight binders. And in summary, we actually found a number of these peptides that were effective inhibitors of the DUB module. So inhibition means we're not showing cleavage of ubiquitin here. The most effective of which actually had a 20 nanomolar IC50. And not only that, but it actually is quite effective in cells in actually boosting the rate of ubiquitination. These are untreated cells, and these are a number of inhibitors. This is our most effective one here. So you're really getting greatly elevated levels of ubiquitination, and it's quite specific. So this is the inhibitor, which is showing you that it basically doesn't work on a number of these human deubiquitinating enzymes, and we've actually recently tested on others as well. So we're excited about the finding. We are returning to X-ray crystallography to try to crystallize our complex so that we could understand the mechanism of action. But we're excited about being a phase now where we are moving forward and trying to take our different structures and using the insights to identify inhibitors, even if our first hypothesis actually doesn't turn out to be correct. The important thing is that we're finding these inhibitors and that they could potentially be effectively developed into drugs. So I just want to thank the people responsible for the work. We took this picture a while ago in pre-COVID time, so I've had to add faces, but in particular, I want to call out Evan Warden, an incredible postdoc who's about to start his own lab at the Van Andel Institute, so watch out for more work from him. Nick Hoffman worked on the Dot1L project. Mike Morgan did the work on drug discovery, as well as actually solving earlier structures of the DUB module bound to nucleosomes. If you are interested in joining our group, we have a postdoc position available. These are the entities that funded the work. And finally, I really want to wish happy birthday again to PDB50 and looking forward to the next 50 years. So thank you for your attention, and I'm happy to answer questions if there are a couple of minutes.
Thank you very much, Cynthia. So we do have time for 1 or 2 questions. So please put them in the chat or unmute and ask your question.
Cynthia, it's David Rose. You mentioned AlphaFold, obviously, on all of our minds, and then you showed some quite distinctive conformational changes, including secondary structure changes. Do you want to, can you maybe comment on that and put those two thoughts together?
Yeah, so we haven't yet looked at I'm probably like some of you. AlphaFold has been very distracting. We spent hours in the lab downloading structures. So I haven't looked yet at the compass complex, but what I do know is I've already seen some people have posted on Twitter the predictions for individual subunits in these complexes that really co-fold have not been that accurate. And so really, I think I'm assuming the next phase of AlphaFold is now going to be co-folding of proteins that belong together. I heard that there's something coming out where there's going to be combinatorial pairing, like of all of the yeast proteins in pairs, or something like that.
That might be coming from the Baker lab.
The Baker lab. Okay, yeah, I'm sorry that I just highlighting AlphaFold and I haven't had even a chance.
Rosetta Fold, I mean. Sorry.
Yeah, Rosetta Fold. So that's really the next exciting frontier. But they are probably doing a decent job of some local predictions of secondary structure, and so it could well be, and we haven't had a chance to look at this, that's the overall like quaternary organization, if you call it that, of the elements is not correct, but that there are local elements that are correctly predicted, perhaps, and those could be really useful in interpreting parts of our maps where we can't see anything at all. So we're really looking forward to doing that, but also looking forward to the software and the predictions moving forward that will do that computationally.
I think you're absolutely right, Cynthia. We've been speculating at the RCSB Protein Data Bank about the accelerating effect that the availability of this information is going to have on structural biology. And you mentioned just one example of the potential impact. Very exciting. So let's thank Christine again, Cynthia again, for a great talk.